Hello and welcome. My name is Marc Diederich. I'm a professor at the College of Pharmacy of Sørøst University, and I lead the molecular oncology lab. So there is one question on this slide, and I would like to take a little time to explain this concept over the next 20 minutes. So basically, a very simple question could be, can coumarins turn cancer cells into innovative vaccines? As a matter of fact, coumarins are natural compounds. They come from plants, and we know them from cinnamon, tonka beans, sweet clover, and so on. For most of the last 50 years, labs like mine studied molecules of this kind as inhibitors of cell signaling pathways of antioxidants. So what I would like to introduce today is the question about using these compounds for alternative tasks like activation of the immune system. So these molecules then can turn into something far more interesting than simple regulators of cell signaling pathways or eliminators of free radicals. So this will be the first part of my talk, and as a matter of fact, in the second half, I will speak essentially about the lab. So for most of its history, cancer chemoprevention rested on a kind of a simple idea, blocking carcinogenesis. So this is a very classical concept, reducing DNA damage and oxidative damage. And as a matter of fact, antioxidants were supposed to protect the genome. This is kind of a passive protection, and we are trying to do something more upstream from the beginning. So our new paradigm is different, and as a matter of fact, we know that our immune system is maintaining what is called immune surveillance. And in order to maintain this immune surveillance, they are reacting whenever the so-called danger signals are produced by cancer cells, whether presented at the surface of the cancer cells or whether they are released by cancer cells. And this then leads to a more active recognition of dying cancer cells. And as a matter of fact, for our research, we investigate three different pillars. So one is involved in the investigation of the BCL2 families, which are regulators of cell death. And we were interested to find out whether we can use drugs that are used to interfere with the BCL2 family of drugs by coumarinic compounds in order to activate the immune reaction. So we study therefore cell death, and one peculiar form of cell death is the immunogenic form of cell death, which is essentially the form of cell death that is activating the immune surveillance, and it is the form of cell death that is, as a matter of fact, actively releasing these danger signals. We use a variety of approaches to target cancer cells, including epigenetic reprogramming by inhibitors of HDACs, including HDAC6, by combined treatments, basically targeting HDACs and additional pharmacologic targets like cyclin-dependent kinases, and also more recently targets within the mitochondria, maintaining the mitochondrial structure, so like the protein OPA1. With this, we try to overcome resistance mechanisms against individual drugs and as such to facilitate cancer cure. So here is in more detail a first research axis, basically the mitochondrial pathway to cell death. There are a number of clinically used drugs that target the BCL2 family, like venetoclax, also called Venclexta, which is inhibiting BCL2. And there are a number of experimental drugs, like the A13 compound here, targeting BCLXL, and the S33 compound targeting MCL1. So this all leads to the activation of the small BH3-only protein BIM, which leads to activation of a cell death mechanism at the surface of the mitochondrial outer membrane called Beck and Becks. This leads then to the release of the cytochrome C by pores called mitochondrial outer membrane pores that, as a matter of fact, lead to activation of caspase 9, caspase 3, and apoptosis. As a matter of fact, cells that overexpress MCL1 will become progressively resistant to drugs like venetoclax, which originally interfere with BCL2. As a matter of fact, it is interesting to see that whenever one of these mechanisms is blocked, nature, and as a matter of fact, cancer cells can indeed compensate by activating a second mechanism. So therefore, this needs to be improved. So as a matter of fact, we need to come up with more efficient mechanisms to trigger cell death. And this brings me to the next slide, which is the second research area. It is an induction of cell death that is immunogenic. How to obtain immunogenic cell death and prevent immune evasion to some extent? Well, we take one of these inhibitors, the A13 compound blocking BCL-XL that I was speaking about on the previous slide, and we combine them with a coumarinic molecule, which is called OT55 here, which was in the original title of my presentation. This compound is known to trigger a very peculiar stress in the cancer cell. So now the situation is changing. This MCL1 protein I was talking about on the previous slide is now strongly downregulated. And other activators of cell death are indeed increased. This leads to cell death and the emission of the DAMPs or danger signals I was speaking about. They are called calreticulin, ATP, and HMGB1. These compounds eventually target dendritic cells, and as a matter of fact, they start activating CD8 positive cytotoxic T cells. So as a matter of fact, by combining compounds, one initially targeting anti-apoptotic mechanism, and then the coumarinic molecule of natural origins triggering a very strong ER stress, we reprogram actually the compensation mechanism of cancer cells, and in particular leukemic cells. We release DAMPs, we activate dendritic cells and CD8 positive cytotoxicity. This can be further stimulated by checkpoint inhibition, the well-known antibody-like drugs, which as a matter of fact allow us to maintain our mice tumor-free. And this is, as a matter of fact, a second important research area. So we are interested in these danger signals. And as a matter of fact, from the, let's say, lab, we move in an in vivo situation and we validate actually our cell-based findings in syngeneic mouse models, and we validate the immunological vaccination. A third aspect will be related to the epigenetic and metabolic reprogramming, as a matter of fact. So this is a little bit how we also move from one aspect to the research to the other. So we started with a target discovery, which would be like a phase 1 of our research. As a matter of fact, this is essentially a work that we do on the computer with computational modeling with partners at SNU. We are also mining public cancer databases. So as we work on leukemia, acute myeloid leukemia databases are very interesting to us. Then a kind of an important part of our research is the discovery of the mechanisms, because it is not only to discover targets, you have to explain the mechanism of the target. As a matter of fact, recent mechanisms over the last year or so was HDAC6, a very specific form of histone deacetylases, which is located in the cytoplasm. It is regulating autophagy and the stress response, and it is tightly connected to the ER stress I was talking on the previous slide. I already mentioned OPA1, a mitochondrial protein that maintains mitochondrial integrity. Also targeting OPA1 leads to metabolic reprogramming, and as a matter of fact, this was very strongly affecting the capacity of cancer cells to survive. Eventually, we translate this also by combination treatments, where we clinically use the cyclin-dependent kinase inhibitors together with experimental histone deacetylase inhibitors, and as a matter of fact, we investigate this in acute myeloid leukemia. We also do combination treatments with tyrosine kinase inhibitors, like the fourth generation asciminib tyrosine kinase inhibitor, which is used in chronic myeloid leukemia. This research then leads to publications, of course, and it leads to a number of research metrics. For example, together with my researchers and students, I came up with over 320 research papers. This leads to an H index of 90. We obviously supported by BK21.4, and here are, over the last year, a number of recent papers in a higher impact factor range, which allowed us to achieve the PhD thesis here at the College of Pharmacy. So altogether, we work starting from patient cohorts, moving to profiling, to target validation to some extent, to mechanism investigation, and eventually to in vivo immunology, if I would have to describe the 4 steps that the PhD and master students undertake in my lab. So we are a small research team. We are about 9 people. We take advantage of all the core platforms of Seoul National Universities, and we collaborate with teams both at SNU, at the SNU hospital, but also at many research teams many research teams, as a matter of fact, abroad. So these research teams abroad, for instance, would be in Luxembourg, would be in France, both at the University of Lorraine and also in Paris, in Italy, in Rome, in France, in the United States, and in Switzerland. We collaborate essentially also with chemists at SNU, but also in Portugal, in Norway, and as a matter of fact, as a researcher and as a professor, I'm also part of kind of an extensive number of editorial boards. Now, altogether, our main forms of recent discovery is this form of combination treatment, as I was mentioning before, including a natural compound derivative to better understand how leukemia cells die, to develop a gene in silico AI-based gene scores based on the immunogenic cell death, to better understand the mechanism, and eventually to create new forms of vaccination. So I progressively come to the end of my presentation. So here are the names of the researchers who recently worked in my lab. We have national and international financial support, together with our collaborators I already mentioned, and also the specific Korean funding schemes that supported the research that I described. So here we have the research team in our recent lab lunch to some extent, where all the students are invited. Also you, if you are interested to join my team, you are free to come over to my office at the building 21, room 218. So you might have questions and we can further discuss. So the lab is open for undergraduate students, master students, and also PhD students. So thank you for your attention.